Good morning, my dear students. Now, in this class, we are going to discuss about cell-mediated immunity. In last class, we have discussed about cell-mediated immunity, that is T lymphocytes is responsible for that. Since T lymphocytes could not able to find out the circulating antigens, it takes the support of MHC. MHC stands for major histocompatibility. T helper cells will take the support of MHC2, T killer cells will take the support of MHC1. The main intention of the cell-mediated immunity is it involves in destroying the infected cells, cancer cells and rejections in transplantations. So, this is the major functions. What are they? Cancer cells can be killed, viral infected cells can be killed, and transplantation time to protect the organs, that antigens also can be killed. Coming to the humoral immunity. Humoral immunity means what? Which is circulating immunity. Some antibodies, some B lymphocytes are modified into plasma cells and memory cells. These plasma cells secrete immunoglobulins. Because on B cell, on the surface, they have a binding site called as antigen binding site. It can easily find out an antigen, and immediately it will tell to interleukins to find out whether it's an antigen or not. Yes, it will confirm it, and the information comes. Then it will how much it is, how much antibodies has to be released. Everything will be calculated by these B cells. As an antigen comes, this antigen is this, antibody is this, it's an antigen and decrease the antigenicity of an antigen and gives the immunity. Memory cells live long periods and exhibit a very quick response towards the same antigen in subsequent exposure. How are antibodies are useful? Here, antibodies are useful in different ways. How to kill an antigen? There are so many ways to express anything. Like that, antibodies can kill an antigen in so many different ways. First is neutralizing the antigen. How? Antibody goes and decreases the antigenicity of an antigen is called as neutralization. Come to agglutination. The antibodies which are not in good condition, they will go think this is an antigen, and this antibody will go and stick to that antigen and keep on increasing the antigenicity. Some cannot move, some will be very circulating. With the help of their flagella, they will, by their flagella, this antibody, some is moving like this, suddenly it will be stopping. Not because of the high pressure on the body, ultimately the antigen can kill by themselves. Causing opsonization of them. How is it done? Opsonization means what? It's a type of phagocytosis. All the antibodies will go and stick towards the antigen and make it into pieces and eat it. That is called opsonization. Next, activating the complementary system and phagocytosis. It will, once an antigens are there, antibodies will find it out whether it is an antigen, and it will activate the innate immunity like second line defense, then third line defense. Systematically it will releases. Once for second line defense it comes now. First line defense only failed only, the antigen can enter into our body completely. Then second line defense will take the responsibility like interleukins, interferons, natural killer cells, etc. Okay, and polymorphonuclear leukocytes, monocytes will take the responsibility. After that, then finally third line defense will come into the action and kills the antigen and precipitation of soluble antigens. Okay, how this can be done and how the immunity can be increased? That is by vaccination and immunization. A diamond can cut a diamond. a microorganism causing an infection, so the microorganism is causing an infection, so the medicine is also there from the microorganism. That is what a vaccine is. It is the process of development of immunization against a particular disease is called as the immunity. How can they do? One by toxoid, another one is by vaccine. What is meant by a toxoid and what is a vaccine? Toxoid is nothing but the metabolic waste of an organism, like excreta or urine, that is converted into toxoid by the treatment of formaldehyde is called as toxoid. There are two toxoids, and diphtheria toxoid, tetanus toxoid. After the conversion of a metabolic waste into toxin into toxoid, and again purified with aluminum phosphate or potassium phosphate, and then sterilized or keep in fridge and then take. Okay, that is what a toxoid is. Whereas vaccine, it can be either inactivating it and taking the, after they die, from there we are taking the antigenicity of it, or by live we will be taking it. By live, when it is in living condition only, we can kill it and we can do it. Okay, and getting the antigenicity of it. This is how vaccines are prepared. There are two types of vaccines. One is killed vaccine, which is also called as inactivated vaccine. Example, polio vaccine and cholera vaccine. Whereas attenuated vaccine, which is called as weakened vaccine, example is smallpox vaccine. Edward Jenner has invented the vaccine for smallpox. The process of administration of vaccine is called as vaccinization. Vaccines are produced by using this approach along large scale production, hence there will be a greater variability. How this vaccine is prepared? BCG vaccine, which I have discussed in the last class, they will collect the bacteria from a person who is having TB. They'll scrap it and they'll culture in a media like agar media or any broth and keep that BCG bacteria in the broth and keep it in an incubator and ultimately grow because bacteria undergo binary fissions and multiple fissions, and n number 20 millions of colonies are prepared. Then we'll be filtering it in a very particular process, then taking that antigenicity, means that antibody, that antigenicity of that vaccine, and keep it in fridge and cool it. Vaccines are even given by injection or by oral drops. Lifelong immunity can be provided by single dose or by giving by 2 to 3 booster doses. Majority good old land is 2 to 3, but now single dose is majorly enough. For babies, 2 to 3 doses are required. Why, sir? Because they don't get immunity via food. But for adults, the immunity can be get via food. That is why it is called as natural active immunity, lifelong but slow. But passive, artificial. Vaccines and toxoids comes under. What is the difference between inactive vaccines and activated vaccines? Inactivated vaccines, as the microbes do not multiply, means it is a killed vaccine, booster shots are required periodically to reinforce the immune system. So dead vaccine, dead virus or bacteria or whatever, will take it. From there we'll take a single dose completely. That is what it is. Booster shots means what? That is why once polio, every year polio day will be celebrated. On that polio day, everyone has to take polio drops. That is what inactive vaccine ki, live vaccine ki variation. Whereas attenuated vaccine, that is a live vaccine, microbes can multiply. Ultimately, a single dose. BCG vaccine once will be taken after the delivery. Again, we will not take it. Why? Because that will be increasing its resistance in the body. What are the uses of these vaccines? Vaccines are just prophylactic measures which tells about an inadequate immune response of the host immune system, and the host develops antibodies. The immune system may not effectively defend the body from the pathogen immediately. With this, the topic immunity is completed. Now, what is the thing is primary lymphoid organs and secondary lymphoid organs. What are the primary lymphoid organs which will majorly takes a part in providing immunity is bone marrow and thymus gland. Whereas secondary lymphoid organs which are supportive, like Peyer's patches, like malt full form, mucosa associated lymphoid tissue. 50% of lymphocytes are produced from that. Then abdominal tonsils, etc. These are the things which we will be learning. In the next class, we are going to discuss about immunity disorders. Under this immunity disorders, we will be learning about hypersensitivity, we will be learning about autoimmune deficiency disorders and immunodeficiency disorders. Thanking you. Signing off, Mahesh Babu.